The fun and inspiring part and invigorating part of Summit and just life in general at the U of M is that we have rich intellectual treasures that are across disciplines, across our 19 schools and colleges, and the people that care about privacy and security are not just limited to folks we hear in the College of Engineering and in law. Rather, we have faculty in our School of Medicine and our School of Nursing that equally care about protecting personal information in research contexts and other contexts. So please welcome Professor Ivo Dinov. He's a professor of health behavior and biological sciences in the School of Nursing and a professor of computational medicine and bioinformatics in the medical school. So Ivo, welcome. Thank you.
Thank you. It's a great pleasure to be here. It's an exciting conference. I really enjoyed what I saw so far. So what I'm hoping today is to take you on a little journey that attempts to balance the notions of privacy, security, and utility of information. So in this talk, I'll present some of the work that my lab is doing, graduate students and postdocs are doing in my lab, that has to do with enabling sharing of sensitive information through statistical obfuscation. So the talk is going to be naturally divided into three components, a beginning, a middle, and an end. And we have to go through all of these to get to the end where the actual beef is. But very broadly speaking, in the beginning, I'm going to define what we mean by big, heterogeneous, complex biomedical and health data. I'm going to provide some of the characteristics. We have come up with definitions of what these things are. Then I'm going to define some of the techniques. I'll mention two of the standard classical approaches that are used that would allow you to share sensitive information that has health-related elements in it. And then I'll present the foundations of our DataSifter algorithm. And then the third part is going to be where we actually take some examples of data that's been desensitized to actually do fairly interesting analytics that are not possible without techniques and strategies to actually enable the data sharing and the computing on that sensitive information. So, before I start, I want to draw parallels between what are data and why are we looking at data. We're not looking at data just for the sport of it, right? The data is essentially a proxy of certain processes that we want to have deep understanding about so that we can actually make inference, prediction, forecast, and so forth. So on the left panel here, you can see that the natural processes, they're very complex. They're most of the time not observable. And on the right-hand side is most of the time what we actually deal with. Very sparse, small data. There is pros and cons to both of these. And where we live right now, we're going to be right here in the middle right up here, where we're going to deal with these larger data sets that have a little bit more complexity. They come from multiple sources, but they present different challenges. It's not anymore, are the data representative? Are they having the same assumptions valid for them? It's more of the nature of the aggregation, the computing, the modeling, and the analytics. So for the past 3 years or so, we've looked at many, many dozens of different case studies, is what we call them. So these are different exemplars of complicated biomedical and health data sets, and we've tried to look at the characteristics that are common for all of these across the different case studies. And these are the 6 or 7 characteristics that consistently come over and over again when we look at one of these. The first one is obviously the big data indicates the size is large. We all know that. But that's not what the big biomedical data is, because sometimes it's actually smaller in size but complex in its intrinsic format. The second one is complexity. Some of these data sets, they have a lot of incongruencies. They have file format heterogeneity and all kinds of things that make them difficult to integrate, difficult to process, model, visualize, and so forth. The data are oftentimes incongruent. They can have different sampling rates. They can have different measurements. All kinds of complexities arise that make them incongruent. Oftentimes, they're multi-scale. You can look at the same phenomena through different prism, either at 30,000 feet, 20,000 feet, or down on the ground level, and you're looking at the same topographical features that you're trying to model. Well, how do you actually analyze data that's intrinsically hierarchical or that's multi-scale? That's a tough problem. Most of the data comes from multiple sources. It's not just the imaging, it's not just the genetics, it's just not the cognitive assessments that physiologists and neurologists do. There is biospecimen, there genetic and epigenetic, hereditary, and all kinds of other factors that come into play that make the data very difficult to process. Most of the time, data has an intrinsic longitudinal dimension to it. It's acquired across time. That presents problems. And lastly, but not least important, is the fact that it's never complete. So these are the common 7 dimensions of what we call big biomedical and health data sets. So typically, a case study that has some of those, maybe not all, but some of these characteristics would not be something that's simple to embed directly into existent computational and statistical modeling platforms. So let me just show you one example of a multi-scale, multinomial neuroimaging data, and if anybody is interested in, there is an online app. You can, right now, as we speak, go and dial the address that's going to show up on the bottom to see how this thing works. So here is a resolution for each of these, kind of shows you from low to high resolution. As you move about and acquire additional information, you can actually go from virtually nothing down to a place where you can actually read some information about some of these brains. So here is the URL of the app. This is something that we developed in the past year or so that allows us to look at extremely complex data set, and I'll show you some examples of this later. But think about tens of thousands of subjects having multiple thousands of clinical assessment measures and enormous multi-model imaging data from which we obtain another set of thousands of derived neuroimaging biomarkers. How do you make sense of this data? Well, the way we do it is we've developed various strategies that will allow us to reduce this hyper-extreme dimensionality of the data into 2 or 3 dimensions, and hopefully in those lower dimensions, we can actually identify associations between clustering, computed derived phenotypes, and clinically observed phenotypes. So being able to look at data multi-resolutionary is of great importance. So that's the first part, the motivation. So I'm going to go now into the middle part where I talk about the techniques and the methods, and there will be some technical components, but mostly I'll try to give you the gist of how these things work. So the 2 existent methods that allow us to share data without compromising personal or sensitive information are, number 1, epsilon differential privacy, and number 2, fully homomorphic encryption. So for both of these, we have slightly different goals. They do have pros and cons. There is no free lunch, so you have to compromise. You trade off pros and cons depending upon your specific situation. So I'm going to go very quickly and summarize how differential privacy, for example, works. So you start off with a bunch of categorical or numerical features or variables, and then you have a list of cases. So these are your individuals or instances of the process that you're observing. So differential privacy relies on adding noise to data in a clever way so that you don't change the first order characteristics. In other words, you preserve the mean, the standard deviation, you preserve various characteristics of the original data, but at the same time, you look at the original and you look at the noise-corrupted data set, and they look very different, right? So, and there is a mathematical way to formalize what this means. So it's basically defined in terms of these privacy protection methods. You look at functions that have certain characteristics in terms of probability measures. You define, and there is many different algorithms that obey these epsilon differential privacy criteria. But just to give you an example, there is many of those machine learning algorithms that have been previously studied. There is also the basic principle is that you have a data set, you're going to do something to it, but before you release it in its native state, you're going to be corrupting it with certain type of stochastic noise. And in this case, the most common one is Laplace noise, which is a double Gaussian distribution with certain dispersion and trivial mean. The fully homomorphic encryption works in a slightly different way, and it's a very, very mathematically elegant framework of doing the analytics. So you start off with having, a data governor that is in possession of some powerful data, some data that has a lot of signal in it, and they want to be able to do certain analytics. So F is this analytics that they want to do, but the data governor is rarely the one that actually does the analytics. So what they're interested in is they're interested in encrypting the data, sharing the encrypted data in such a way so that whoever is the analyst, whoever is the user of this data, can design some of these, they're called commutative analytic evaluators, that would be able to do the analysis on the encrypted data, and then they generate some kind of an inference. This F of the encrypted data is actually my inference, but the key in this is that we want to have this diagram be commutative. So in other words, if I go ahead and get the raw data and apply the corresponding analytics on the native data right up here, and then encrypt that, I should be getting exactly what was obtained by analyzing the encrypted data. So the notion of analyzing an encryption should be commutative. And any function that satisfies this is called fully homomorphic encryption. Now, obviously, there is many, many different ways to do that, but this is a clever way. The only problem with this strategy is that in the absence of knowing exactly what the core analytical processing method is, it's very difficult to argue what is the corresponding commutative analytic evaluator. So the simple ones are easy, right? If you're going to be doing first, second order statistics, these are not hard to do. What is hard to do is to do non-parametric inference and various other complexities. So we have looked at this problem simply because we have requested multiple times from the University of Michigan Health System access to the electronic medical records, and we've been told no. So our team decided to provide a mechanism that will allow the data governor to actually share the data by trading off the risk of re-identification of individuals or extracting sensitive information and preserving the utility of the data. So roughly speaking, this is a statistical obfuscation method. The way it works is it iteratively goes through the entire data set. I'll show you a diagram in 1 second, and manipulates the data in a way in which it tries to preserve the joint distribution of the original data, and at the same time, to some extent, it guarantees preservation of the privacy. So here is a schematic of roughly, and you can see this. This is all open source. You can look on the Google on the GitHub partition for the soccer project. So we allow the data governor essentially one parameter, eta, that allows them to vary from releasing the data on the clear at one extreme, all the way to the other extreme where they release completely synthetic data that has nothing to do with the original. And anywhere in this continuum, the data governor can actually specify what specifically is the trade-off between minimizing the risk of individual identification and maximizing the utility of the data, right? So obviously, nothing is as powerful as the original data, but if you can't have the original data, what kind of data can I have that kind of has the same energy, but balances up the notion of, as I said in the beginning, preservation of sensitive information and preservation of utility. So here's a diagram of how this process works in practice. You have Jane and Joe, two users that are interested. They're completely independent of the data governor. They have defined their initial queries. I want this type of data. Joe says, I want this type of data. They send the request to whoever is the data governor. The data governor translates that request into some kind of an SQL or no SQL query. They put it through their electronic health record or our database, and they extract all the matching cases. Now, note in this schematic how there is missing data, right? That's perfectly fine. And then this data, the data governor, not these guys, the users, is sending this to the DataSifter, which does the number crunching. Well, first of all, the data governor decides, well, how trustworthy are Jane and Joe? And then they specify this eta parameter in between. And they start this iterative data sifting of the data. And it goes through the process. You know, here is the original data that got fed in, that got extracted from the database. So we go ahead and obfuscate on the fly randomly selected cases and features. We go ahead and impute these things, and then we repeat this multiple times in such a way that we try to preserve the distribution at every one of these phases. We try to preserve the distribution of your data. And at the end of this process, I have a complete data set that's being sent back to Jane and Joe. Now, this data is obviously changed. It's altered. It's adulterated. And then what they do is they actually design their workflow protocols. They pipe the data to something, and they get some kind of a result. So that's the mechanism. That's, in a nutshell, how the DataSifter works. So let's talk about, does this thing really work in practice? So in this diagram, so I'm going to go through several validation phases to kind of argue that what we are doing here is indeed nicely balancing the preservation of the energy of the native original data, and at the same time changing it enough so that re-identification of individuals is kept at a very low risk. So what you're looking here is, you're looking, remember this ADA parameter that I said the data governor controls depend upon the trustworthiness of the requestor of the data? So this is, we've actually go ahead and specified 5 different levels of this. So releasing data on the clear, little obfuscation, medium level, large level, and releasing completely synthetic data for the most untrusted individuals, right? So these are 3 different levels. What you see on the vertical axis is something that we call percent identical feature value. So this kind of tells you what proportion of the signature of, in our case, the clinical information of the individual gets sent back to the requester at any one of these obfuscation levels. And you see in the beginning, if you release data on the clear, obviously you're sending all the information. If you do small obfuscation, about 80% or so of the features are unchanged, and as you increase the level of obfuscation, fewer and fewer features get unadulterated, okay? And as you go to large obfuscation, the vast majority of the data elements are changed, but remember that in that change process, we try to preserve the distributions. That's the key component here that allows us to keep the energy at the same time we do the sifting. So in this specific case, we used synthetic data for this specific validation, where we actually know the exact characteristics. In this second example, we are using a slightly different way of validating. What we're going to be looking at is we're going to look at various models. Now, suppose I release the data to you, and you say, okay, I want to have a model-based or a model-free inference. And the question is, how much my inference would change in either of these situations if I work with the raw data, slightly obfuscated data, moderately obfuscated data, or severely obfuscated data? That's a reasonable question to ask, right? How is the inference, the forecasting, altered by that process? So right up here, you have several different situations for binary outcomes, for binary classification, for polytomous or multinomial classification, as well as for the continuous variable. We have different colors kind of indicating. And here is again, the true positive rate is on the vertical axis here, and the false positive rate on the vertical axis in the second diagram. So we can see that you have, a lot of things are being preserved there. You don't change, obviously, the inference early on, and as the level of obfuscation increases, the amount of true features that you're detecting in the data is naturally going down. So I'm compromising the utility of the data by extreme obfuscation. And similarly, right up here, the false positive rate, if I'm not doing any obfuscation, the results are not going to be different. I'm releasing the data on the clear. So you don't have any false positives, but as you keep increasing the level of obfuscation, obviously more and more false positives are going to show up in your analysis. So this is all nice and dandy. It's using simulated data. Now let's look at some real data sets. So in this specific case, we're using a fairly large data set called a BITE, Autism Brain Imaging Data Exchange. It has about 1,100 pediatric cases with over 2,000 features that we have. And we're looking again now, this time instead of horizontally, this time the obfuscation level is on the rows, as you can see. And now this is just for one individual. We're kind of randomly picking the 22nd individual in the collection of 1,100 or so. Picking a 27 individual and just picking some of the many thousand features that we have to see what is the effect, what is changed, and what is not. So this kind of empirical evidence. This is very, very empirically driven. So first of all, the clinical assessment of this individual at the original data set is patient, gender is male, age is 31.7, the type of imaging scan that's acquired is sagittal, and it's got an IQ of 131 and various other characteristics, okay? And then as you go between the no obfuscation, obviously it should not be changing anything in this data set. But if you do small obfuscation, notice how there is very little that's been done here, except a little bit of a change over here. At median obfuscation levels, now all of a sudden the IQ is changed. The IQ is changed. You go from 131 down to 111. And then some of the other metrics are somewhat changed. And as you go all the way down to the bottom, you see even the clinical diagnosis of the individual at the extreme obfuscation level, you change the clinical diagnosis, which is obviously quite extreme. Now, for this specific individual, the original and the obfuscated may be different, but I want you to think about this population-wide. Because typically, when you release a data set, you're interested in the overall energy, the overall information that it contains, not the information that a single case contains. So for this specific very same data set, what we did is we did a clinical assessment trying to predict the patients, the autism spectrum disorder subjects, from the asymptomatic age-matched controls. So we are having, again, the different levels of obfuscation, from no obfuscation all the way up to complete obfuscation. And again, the vertical axis here shows the percent of identical features that are unchanged. And you can see, again, what we saw before, a fairly stable initial for small levels of obfuscation. You don't change a whole lot, but as you increase the level of obfuscation, most of the features do get altered. And on the right-hand side here, we have the prediction accuracy. And you can see that, again, we are very, very accurate early on, and that accuracy tends to drop down as you, and now obviously in this case, accuracy of 50/50 is a completely random assignment of a computer-derived phenotype. So the 50% level is the additive no, no. So this again kind of shows us that we can that we see what we expect even with real data sets. So we've got this technique now. So we have a mechanism of getting data that's sensitive, and it doesn't have to be held data, right? Any data that comes from IRS, from other locations, insurance data, could be CMS, Medicare data, or any other information that, obviously I'm talking about social security numbers and names, these are obviously removed. Nobody cares about this. The question is the sensitive data that includes phenotypes that can be used to actually search through the database, and if you find a phenotype of an individual that you know is there, the question is, can you say that they're part of the database or not? That's the question that we are trying to address here. So now that we have a mechanism of obfuscating data, what can we do with data that's large if data governors are obviously willing to share their data subject to that statistical obfuscation process? So the first example that I want to show is, and again, this is something that you can try online as we speak. This is something called the data dashboard. So it provides a web service for combining and integrating multi-source data sets that come from completely independent sources, aggregating them using some anchoring features, and then doing dynamic, on-the-fly visual analytics of the entire data set. So you can see a collage here of different charts. In this specific case, we have used 3 data sources. The data came from the Bureau of Labor Statistics, the Census Bureau, and the Center for Medicare Services, CMS. We pulled data from these for the year 2011. We integrated it, and what we can do with this is we can actually look at ways in which this data can be visually interrogated. So this example here shows two examples. The first one is right up here where we have a chart of the population density, for example, by state. And then you have a histogram of the labor force, a histogram of the average cortical surface that was in the background here. This is pie chart of the race distribution, levels of education, and all kinds of things. And now these are all dynamic maps that will allow you to manipulate, make a selection in any one of these charts is going to dynamically morph the other ones and show you what is the effect of subsetting or stratifying on one feature, what's the effect on the other features that you have in the collage of images. So here is an example also that we have done with our very own University of Michigan Udall Parkinson's Disease Center, a very similar thing. We can put the subjects together after the DataSifter has been applied, and we can actually share this thing on the web so that others can actually look at what we have and try to formulate, for instance, various hypotheses about what are the factors that can be used to predict falls, physical falls of individuals that suffer from Parkinson's. So, a little bit, two very quick definitions. The first one is about data science, and the second one is about the predictive analytics. So data science, as we all are probably familiar, is this new fourth pillar of scientific discovery. It's a new, extremely transdisciplinary area. We have 19 schools and colleges of the University of Michigan, and they're all involved in the Michigan Institute for Data Science, one of the premier institutions for data science in the nation and the world. So all 19 schools and colleges. So that's the extreme transdisciplinarity of the field. It always deals with enormous amounts of complex data, just like the 7 characteristics that I showed early on. On the other side, predictive analytics is a process of utilizing data science approaches, using advanced mathematical modeling methods, computational statistical techniques, web services, novel technologies, and so forth, to actually forecast and predict a variety of outcomes. In our cases, prognosticate the progression of the disease, and so forth. So this little figure shows the typical workflow when we have biomedical and health data going from the raw big data all the way up to the action that is taken based on this data. Now, somewhere in the very beginning here, after the big data is collected in the leftmost column, the raw observation bits and bytes are collected. That's where the data sifter comes in. The data is obfuscated, and from this point on, the field is wide open. You typically extract certain information, for instance, from the K space, from the K domain. You can reconstruct visible two-dimensional images that can be stacked together to form three-dimensional solids that represent human brains, for instance. Out of these, we can extract cortical surfaces, which are unobservable phenomena. There is no way to observe natively, intrinsically, the cortical surface of an individual, non-invasively, obviously. If you start poking somebody's head, in surgery, that's a different ballgame, but you get a patch. Non-invasively, we have to reconstruct these things, so we develop mathematical methods. So that's what the information column here is. And then this leads to acquisition of knowledge in terms of, for example, these things can be parcellated when it comes to the two-dimensional or three-dimensional solids. The two-dimensional manifolds can be partitioned in such a way so that we can model as triangulated surfaces each of these regions. And once we have this, I can actually compute a variety of morphometry measures that have very localized representation of brain physiology. And at this point, once I have these signature vectors, they're going to be completely homologous between individuals, and we can actually do computing. You can't do computing on raw brain scans because they're extremely heterogeneous. Remember, I told you at the beginning, the congruency, the homogeneity, these are important things to have. They're not present, even if the same scanner acquired the data. Human brains are very different. Some people miss foci. It's a mess. It's very difficult. But once we have the information column here, we can extract the knowledge column by using sophisticated mathematical tools. And once you have all this now, you're ready to do forecasting, to do action, to do policy, decision making, whatever. And it's all predicated on data sharing. It is very important. It is very important, and I know it's hard, and people are attached to their data. Their data is their property, it's their baby, it stays with them. It's a problem. It's a problem. And we are risk adverse, and many of us here are aware of the extreme situations of risk aversity. Yes, you're going to keep your data, but you have to keep in mind two very important things. The world is moving at the speed of light, and the second thing is that the value of data exponentially decreases with time. Hoarding data hasn't helped anybody and won't help anybody in the future. So we're shooting ourselves in the foot by not sharing the data. So this predicament is very important, and here is a little bit more of a specific rationale of why data should be findable, accessible, interoperable, so that we can pool data together from multiple sources and actually make some sense, and reusable. It's very important. Data has a lot of energy. For those of you that might not have heard, the Kreider's law is the parallel to Moore's law for data storage. The Moore's law says that our computational power doubles every 18 months. The Kreider's law says that the amount of data that we collect increases every 12 to 14 months. And you can imagine what happens. Both of these exponential laws of increase are going to collide at some point, and we're going to be collecting more data that we can process, and disabling intentionally or unintentionally preventing sharing of data is a huge problem. So let's look at some specific cases now. One of the very debilitating diseases is called amyotrophic lateral sclerosis, Lou Gehrig's disease. The most famous individual was Stephen Hawking, passed away early in March this year. We had looked at a European consortium that had collected about 8,000 individuals in a clinical trial of ALS patients. And they had, as you can see here, many thousands of clinical assessments for these individuals. What they were interested in, and this was an international dream challenge that was put by Biosciences, you're given the baseline information for these individuals and a 3-month follow-up, so 2 time points, and you're asked to predict the state of the individual 12 months down the road. So very, very difficult challenge, which would not have been possible unless people had gone through some kind of a process of ensuring the privacy, security of the sensitive part of the information. But the data was openly shared, and we had acquired the data, and we had looked at predicting exactly what the challenge says. We have a couple of papers on this. So the data was complex, and it had very weak signal to noise, which means one of two things. Either the physicians are not looking at the right place when they collect these 2,400 clinical assessment measures, they're not looking at the right place, or the disease is extremely heterogeneous, and it's not one gene, it's not one environmental factor, it's not one, or a handful of factors, it's multitudes of factors that need to be teased out to find out exactly what causes and how far is the disease going to progress. Some individuals, like Stephen Hawking, live for 50 years with the disease. Most individuals decay very rapidly after diagnosis and are not as lucky. So in this case, we had to develop 3 years. An army of students and colleagues have worked on this project, and we had designed a fairly complicated workflow protocol that starts right up here at the very beginning with the raw data, millions of observations. We dealt obviously with things like missing values. We split the data into testing and training data sets so that when we do the validation at the end, we're not fooling ourselves. We want to have an external validation of the efficacy of the methods that we're developing. And then we go through a whole bunch of other processing tools, fitting linear, model-based and model-free methods. We do forecasting. And at the end, right up here is where we do the assessment. And the assessment, we've done many of these. I'm just showing 4 specific techniques here, from a model-based to 3 model-free methods, machine learning methods. The Bayesian adaptive regression tree was the one that did the best. We can identify which of the 2,400 clinical features that physicians have acquired are the highly powerful ones to be associated with individuals either at 12 months rapidly decaying or mostly remaining stable. Okay, so that was what we were trying to predict. Mostly stability versus rapid decay at 12 months. So we kind of know the rank order of the features, but if you look at these measures, the correlation, for example, how close was our prediction of the decay relative to the actual for the individual decay observed at 12 months, this is only below 0.5. It's not great, and that's the best method that we can do. So the problem is very, very difficult. So then what we said is we beat that problem with every method, technique, or tool known to the human race, and we couldn't get prediction that's better than about 70%. So this came to a realization that the unique clinical phenotypes that the physicians are interested in forecasting are not predictable from the set of features that we are given. So then what we said is, how about we look at the data in its holicity without a prior assumption about the clinical phenotypes. We're going to interrogate the data raw. We're going to let the machine decide what is important here. So we put this through multiple different clustering methods to see if we can obtain derived phenotypes, machine-computed derived phenotypes. And if we can do that, how reliable are our phenotypes is the question. So right up here, again, following the DataSifter, we do all kinds of fusion, harmonization, cleaning, and then here is the model fitting. And then ultimately, the algorithms kept coming over and over again with 4 classes. The algorithms kept saying over and over again, there are 4 groups of individuals in your 2,500 cases. So we said, but what are these four groups? What do they mean? Well, the first thing that we had to do is we had to look at, well, how consistent are these when you run the algorithms multiple times or use a different algorithm? How much consistency is there in the clusters? And you can see that these are pretty high consistency levels. Individuals that ended up being in the same group over and over again was very high. And that's a good sign, because that says that there is energy in the data that can be exploited. The second column tells me, well, how much variability was there between individuals? So these are the individuals that tend to jump from one group to the next when you regroup the cases. Very, very tight. The clustering size, that's the 4 clusters are approximately of the same size. That's encouraging. And the last one says, the silhouette plot, let's not worry about it. It's a measure of reliability of the cluster. And these are above 0.5, which simply indicates these clusters are great. So now we present this to our clinical colleagues, and they say, this is all very, very nice and dandy, but what's class 1, what's class 2, what does it mean, how do I interpret it? And now you have this set of thousands of features, and you're trying to find out the features that discriminate predominantly between the 4 clusters that we have. So I'm just zooming in here on a few of these, just to show you that when you have 4 groups, and you want to find out what features explicate the between cluster differences, you got 6 possible ways to arrange these 4 groups into pairs, where the order doesn't matter. The number of combinations of 4 elements taken 2 at a time is 6. So we're looking at these 1 versus 2, 1 versus 3. Right up here is the second cluster versus the 4th cluster. And then some of these features that discriminate, some features discriminate between any pair of clusters, and some discriminate only between certain pairs. So imagine now you're the physician, and I'm showing you this diagram. What's going to be your conclusion? Is this useful, or is it just hype? The important thing here is that we are demonstrating empirically very strong evidence to suggest that the problem of univariate clinical classification for heterogeneous disease is not solvable. What is solvable, however, is solving the heterogeneity of disease problem, because now we are telling you that there are absolutely 4 clusters in this data set. And I can tell you exactly who is in each cluster. But if you want to know how does my features that I collected, and whether or not these are the right features, I don't know. Right? If any other data is available, we'll take it, obviously, and we'll reanalyze it. But we can point out very specifically which of the features discriminate between the clusters. So the power is that we can do this multivariate classification. And we went one step further. We said, fine, we've got these 4 clusters. Now, remember, they live in this because I have 2,100 or so features for every data set. What happens if I get this extremely high dimensional 2,400 dimensional space and I collapse it in 2D? Would the labels of the 4 classes coincide with any structure in that lower dimensional space? And the answer turns out miraculously to be absolutely. So we're using completely independent, highly nonlinear T-distributed stochastic neighbor embedding technique to project this high-dimensional data into a 2-dimensional manifold, and I've got all the points laid out, and now we're going to assign to every one of the points the label of the code that indicated which cluster it ended up with. And you can see the 4 arcs here. Undeniably, there is structure, and the structure coincides with the corresponding computed derived phenotypes. Okay. So one last example of another study which also required data sharing, data sifting, pre-processing, desensitizing data. But it was a very interesting study because now this involved not only one institution but 2 institutions, and obviously this could have been 15 institutions, which agreed to share data on Parkinson's disease, and they were interested very specifically in predicting falls. So there were 4 specific challenges. The first one is, okay, so you've got this data from multiple sources. How do you aggregate them? The 2 studies were done completely independently of each other. So obviously, the type of information that was collected was not congruent. The cases were different. The cohorts that they actually clinically assessed might have been slightly different. So there were a lot of issues that had to do with the multi-sourceness, the multi-site, and all kinds of other things. The second challenge was to identify the salient features that are highly associated with falls in the elderly Parkinson's disease individuals. The third one was to identify and classify the performance, essentially assess how good is this performance. And the fourth challenge was to look at predicting other clinical traits. Parkinson's intrinsically has 2 different phenotypes. The first one is of tremor dominance, the limbs are shaking, and the second one is postural instability, where people kind of freeze and they may fall because of a freeze. So these were the things that we did, that our goals of the study, and then we were able to produce certain prediction of what are the individuals that are likely to fall, and our results were between 70 and 80%, not exceptionally good, but under the circumstances of analyzing univariate predefined clinical outcomes, that's the best you can do. Just to give you a little bit of a perspective here, the fallers and non-fallers, there is no single set of variables that would discriminate them. That's what this diagram says. They're pretty blended if you look at them. And here is the actual results of all this. So we have tested again multitudes of different techniques, and we're assessing different measures: accuracy, sensitivity, specificity, area under the curve. And these are the features that seem to play the biggest role. Again, 70 to 80% is the best that you can do using random forest in this case. I'll just switch and show you this very interesting data that I mentioned early on, the UK Biobank. Look, at the University of Michigan, we can share our electronic health record data. We can. And our team here at the Information Assurance Office, the Data Security, the Vice President for Research, you heard Ravi early on, they're trying. They're trying, but in the meantime, most of us are not going to be sitting around doing finger oppositions. what we are going to be doing is we're going to be looking at data. The United Kingdom has released 500,000 individuals, data for 500,000 individuals, and you got to go through the process, right? You got to have an IRB at the local institution, you got to sign a memorandum of understanding. You know, this is all fine, and that's perfect. We've done that. We got the data, and now we're looking at, you know, tens of thousands of individuals and many clinical imaging, genetic, phenotypic observations for these individuals. So this is a very difficult problem. We have, just to show you an aspect of the difficulty, is this notion of the missingness. The patterns of the data being incomplete is just very crazy here. The missing counts are on the vertical axis, and just for the most commonly observed 1,500 features, you can see that very few are fully observed. Most have thousands of missing cases. These are obviously not missing at random, as you can just see from this diagram. There is some patterns, so there's a lot of difficulty with this data. But we have developed a protocol that will allow us to automatically go through the tens of thousands of subjects in a large-scale distributed computing environment using the graphical workflow to obtain the valuable information out of it and do forecasts. So I will stop here with just one last slide that I have to show to essentially give credits to my colleagues that have participated in this. So this is the funding agencies that have contributed. The team, Simone Marino here from our lab is in the room. That's the students. The website is over there. If anybody's interested in the slides, our motto is, it's online, therefore it exists. Nothing that we do is not available online. Just Google soccer, you'll find these slides. Thank you very much.
It is question time again. Do we have questions for Ivo?
Good afternoon. One of the slides you had talked about, it was the sharing of data, talked about democratization of the scientific discovery process. So I'm interested to hear you talk a little bit more about that in terms of how it's going to work in practice. It does strike me that the statistical obfuscation methodology you're talking about is probably pretty expensive to do for big data sets, so I would think it needs to be done at a place like the University of Michigan that has that kind of resources. So how does it get democratized out to smaller research institutions, to individual researchers?
Yeah, so that's a great question. And I'll tell you, democratization of science has several components to it. First of all, democratizing the notion of data acquisition. Not everybody even has access to cases that they can monitor if they want to acquire data. On the second level is sharing of data, naturally coming up second. The third one is sharing of software code, tools, and services. And the last but not least that you're kind of hinting to is sharing infrastructure. All of these need to come together because you're right. Institutions like the University of Michigan that are rich in their intellectual and infrastructure capabilities, we can do these types of things. But specifically for the DataSifter, we have released the code. It is computationally intensive, but typically you want to separate the data governorship who actually need to do the obfuscation and the folks like us that are methodologists and then the end users. They need to be separated just to avoid potential conflict. So it is a tricky problem, but it requires, government, institution, individual, industry. It does require a little bit of, industry donate cycles, for example. Amazon, Google Cloud, Azure. It's a multi-pronged approach. Good question.
Thank you.
Another question?
So it's actually following that question and your answer. So I am also very much interested in data fusion, but also in practice for data separation for privacy concerns. But you always have to know what the model and what the question will be in order to know what you lose. So in all your beautiful illustrations, you know what the model was, the obfuscation was able to capture whatever you missed. But if you want to separate the entities, you will need to have some collaboration in testing what has been missed. And how do you think it's going to be like kind of a back and forth process?
Right, so Well, that is a tricky question, because if I'm Jane or Joe that got the complete data set from the data governor after the DataSifter or after another process to obfuscate it, I never know. Because this becomes to you the native data. You don't know what the data might have been. And in our case, this typically requires building some kind of a relation where your level of trustworthiness increases, so can they dial down this data so that they can give you something a little bit more powerful. There is no doubt. Anytime you obfuscate data, you're taking away energy. And you as a end researcher user, you never know how much of the energy was taken out. So that's a problem that's difficult to solve in an abstract, generic level. It always needs to be on a specific case by case.
Thank you very much. Thank you for your speech, and thank you for your attention.
Thank you.